Hi everyone, welcome to our second lecture of Chapter 26, Metabolism. We've had one very brief lecture over Chapter 26 already. Today is going to be the wrap-up of Chapter 26, and it's going to be quite a bit longer and more intense than our previous Chapter 26 lecture. Today we're going to be looking at cellular respiration. We're going to be looking at the activities of the mitochondria. Here are our not really attendance questions for the day. How many membranes does a mitochondrion have? What is the substance in the middle compartment of mitochondria called? And when one side of a membrane has more positive charges than the other side, what type of gradient is this? All of these came from either Chapter 2 or Chapter 3. These are going to tie in directly with some of what we talk about today. So it's a good idea to go back and refresh on these if you don't know the answers. So go ahead and pause the video and try to answer these. And let's see if you got these correct. How many membranes does a mitochondrion have? Mitochondria have two membranes, an outer and an inner. We talked about some differences in those two membranes, which are going to be a little important today. So go back and listen to that part of Chapter 3 lecture if you don't remember that. What is the substance in the middle compartment of a mitochondria called? So this is inside not just the outer membrane, but inside the innermost membrane. It's called the matrix. It was that thick jelly-like substance, and we're going to see a lot of activity involving the matrix today. Talking about those two membranes again before we go to the next part. Remember when we looked at mitochondrial membranes, the outer membrane was very smooth and resembled a plasma membrane of our cells. But that inner membrane was very folded, and it had these folds called cristae. Sometimes you'll hear them called rugae. And that folded part of a membrane, remember any membrane when it's folded, typically that's important for increasing surface area. And finally, when one side of a membrane has more positive charges than the other side, what type of gradient is this? That creates an electrical gradient. So we're going to look at some activities of electrical gradients today and then again later in the semester. So make sure you're really comfortable with chemical gradients or concentration gradients and with electrical gradients and the various types of gradients. Refreshing some of what we talked about in the previous lecture and way back in Chapter 2, we're going to talk about cellular respiration. Here's our chemical equation for cellular respiration. Up to this point, every time we talked about it, we said you did not have to know it. But now you do. You do need to know this equation because it's going to be what we're talking about today. And we will come back to the same equation at the very end of this lecture. And I believe after today's lecture, it will make a lot more sense. So even though we can metabolize everything that we eat that is not waste, we're specifically going to be looking at metabolism of carbohydrates because it is primarily carbohydrate metabolism that we use to generate ATP. And that's what we're talking about, is how we generate ATP. And regardless of which carbohydrates we have in our diet, they're all going to be converted to glucose, and glucose is what will enter cellular respiration. So here we have the equation for cellular respiration, C6H12O6, and remember that is glucose, plus 6O2. O2 is just the oxygen that we breathe. Remember O2, that's two atoms of oxygen joined together. That's called molecular oxygen, and this is what we breathe. So carbohydrates in the form of glucose from our diet and oxygen from the atmosphere that we breathe. These are the two reactants that are going to enter cellular respiration. This arrow represents all of the reactions that occur, and the products are going to be 6 molecules of water. So we produce water through cellular respiration, and 6 molecules of carbon dioxide, CO2. This is why we breathe CO2 out. This is where that CO2 that we breathe out comes from. And we generate ATP, energy. Since we are converting between chemical energy in the bonds of glucose, then what we're going to end up with is heat, because remember, it's going to be very inefficient when we convert. Here's our mitochondria. Down here we see that orange, the very smooth outer membrane. We see the inner membrane with those folds, the cristae. This kind of very faint yellow color here, that's the matrix. That's that thick jelly-like substance we talked about. There is some space in between the outer membrane and the inner membrane, this kind of dark yellow, faint orange color. This is called the intermembrane space. And all of these things are going to be very important for the steps of cellular respiration. We're going to talk about what happens at each of these locations, and you are expected to know that by the time we finish this lecture. And one other thing that's in this picture that isn't labeled, all of this white area outside the mitochondrion, what is that? Well, that's the cytoplasm, because the mitochondria are inside of our cells. So if we're inside the cell but outside the mitochondria, this is the cytoplasm. And that's also going to play a role in today's lecture. So what we're going to do is draw the steps of cellular respiration. Each step we're going to draw through. Now your book does have all of these steps. In chapter 26, figure 26.5, 26.6, and 26.8. Those are going to be the steps of cellular respiration. But if you look at those, they can be a little intimidating. So we're going to draw them step by step and talk about the parts that are important versus the parts that you can overlook. And we're going to see what those steps are actually showing us. And I will talk about exactly what you need to know coming from each of those steps of cellular respiration. So first one that we're going to look at is glycolysis. And this is, I believe, figure 26.5 in your textbook. And like I said, there's a lot going on in this picture. So we're going to just start at the very, very top and work through step by step and see exactly what's happening here. Now, in today's lecture, we're going to be talking about things that we learned in the previous lecture. So if in lecture 26, part 1, if you are not comfortable with the terms that we introduced, please go back and rewatch that lecture before continuing in this lecture. We're going to be talking about NAD plus and NADH and FAD and FADH2, all of those things we're going to be talking about without really explaining what they are, because we've already done that. So please go back and rewatch 26 part 1 if you are unfamiliar with those. Now let's back out and go to our whiteboard so that we can draw step 1 of cellular respiration. Now in cellular respiration, there are going to be very distinct phases, and different things are going to happen at each of those phases. The very first part of cellular respiration, even though we're talking a lot about the mitochondria today, happens outside of the mitochondria. It happens in the cytoplasm. So step 1 happens in the cytoplasm, and it is called glycolysis. Glycolysis literally means splitting glucose. Now remember, when we talked about glucose, we saw that it was C6H12O6. And we saw back in chapter 2 that typically it's drawn in kind of a ring-shaped structure, but we're going to draw it a little bit differently. In your textbook, when it shows glucose or several other molecules that we're going to be looking at today, it shows 6 little black circles joined together. And each of those circles represents a carbon. How many carbons are in glucose if it's C6H12O6? 6 carbons. I'm going to draw it a little bit differently. Instead of showing those 6 little black circles, I'm going to show 6 C's, C for carbon. 6 C's joined together. Now, that doesn't mean that all we have is carbon. Remember, we have H12O6 attached to that C6. So there are hydrogens and there are oxygens there, 12 hydrogens and 6 oxygens attached. We're just not drawing them for a couple of reasons. One, it would get really, really crowded. And two, sometimes when we're talking about organic chemistry, the carbon is really what we're focused on. So just remember, even though we're going to be drawing a lot of C's today, there are H's and O's in the background, and anytime that they are important, we will mention them. So this chain of 6 carbons joined together represents our glucose molecule, and that's our starting part. That is our glucose. And our goal here is to make ATP. But something else is going to happen first. Let's start with 2 molecules of ATP. And how does ATP work? How does ATP provide energy? That should be something that you remember from before. It's going to phosphorylate something. It's going to give away a phosphate group. And that's what's going to happen here. And I'm going to show that with a little fish hook. And that fish hook just means something is happening here. And the same thing on the other side. So what is it that happens? We're going to take this 6 carbon glucose and attach a phosphate to each end of it. What does that leave with our ATPs? If we gave away our phosphate, then we're left with ADP. 2 of them. Where did our phosphates go? They went on each end of this carbon chain. Well, why did we do that? If we're trying to make ATP, why did we just use ATP? Well, because in order to split glucose into something that we can use, we actually have to invest a little bit of energy. What kind of energy? Well, energy to split this glucose in half. This middle bond right here, we are going to break it now. And the energy to break this bond comes from those phosphate groups that we just provided. And what we end up with are 2 3-carbon molecules with a phosphate attached. Now, we want to make sure that nothing else attaches to these carbons, and we want to make sure that nothing or that they don't reattach to each other. So we have to occupy these open spaces that we just created. And we can do that with more phosphate. But we don't need to use more ATP. How else can we get phosphate? Remember from our previous lecture, some phosphate is just floating around in the cytoplasm, not attached to anything. We called that inorganic phosphate. Well, that's what's going to happen here. The inorganic phosphate is going to come along and attach to the free end of that 3-carbon chain. So now we have a 3-carbon chain with a phosphate group on each end of it. And we have 2 of those, 1 for each of the 3-carbon chains that we created. Now that we have something that we can easily work with, 2 3-carbon chains, we can start to get to work in harvesting a little bit of energy. Well, remember our NAD+. One of them is going to come in and do his job for each of these 3-carbon chains. What does the job of NAD+ encompass? Well, every time we see him, remember he does the same thing. He's going to come in, take some electrons. Some protons come along too, but his real job is to take some electrons. What do we call him when he takes electrons and he is now carrying electrons? We call him NADH. And the same thing over here, NADH. And remember, sometimes you will see plus H plus because a proton comes along that doesn't really attach to anything. If you want to write that on each side here, that's fine, but it's not necessary. So NAD plus comes along, takes some electrons and some protons, and leaves carrying those electrons and protons as NADH. Same thing over here. And the NADH just leaves. He takes those electrons and he leaves. He's done his job. Now that we've taken electrons and protons, we can go ahead and free up this 3-carbon chain over here and this 3-carbon chain over here. We can get rid of these phosphates. Well, do we want to just let those phosphates go, or should we make use of them? Well, we might as well make use of them. Get something good out of it. And what we're going to have is 2 molecules of ADP and another 2 molecules of ADP are going to come along and grab those phosphate groups. One ADP is going to grab one phosphate, another ADP is going to grab another phosphate. Same thing over here. An ADP will grab one phosphate and the other ADP will grab the other phosphate. So we are going to move these phosphate groups onto these ADPs. The phosphates started out attached to one molecule, and we're going to attach them to a different molecule. What did we call that? Do you remember from last lecture? That is substrate level phosphorylation. And we're going to make 2 ATPs from that. And over here, we're going to make 2 more ATPs from that. So we just made 4 molecules of ATP. We have just seen the first bit of energy production from cellular respiration. Now, we've still got a long way to go. That was just the first bit. So let's go ahead and see what happens up here. We are left with 2 3-carbon compounds. And those 2 3-carbon compounds, each one is called pyruvate, or we could call it pyruvic acid. Pyruvate or pyruvic acid. Those 2 words are kind of interchangeable. They're not exactly the same thing, but for purposes of our class, we can say the suffix ate and ic acid are interchangeable. So that's the end of glycolysis. That's the end of step 1. So let's go and see all of the important things that we will take away from step 1, glycolysis. There's a lot going on there. What do we need to know? Well, you need to know the name and where it takes place. Everything that I circle in green is going to be what you need to take away from each step. So glycolysis, step 1, happens in the cytoplasm. We start with a molecule of glucose, and we end with 2 molecules of pyruvate. We start with 1 glucose, we end with 2 pyruvate. Up here, we have lost 2 molecules of ATP. I'm going to write that here in the middle. We lose 2 ATP. We gain 4 ATP. We lost 2 in the beginning. We produced 4 at the end. So there is a net gain of plus 2 ATP. Does that make sense? We spent 2, we gained 4. So there is a net gain of 2 ATP. And along the way, we produced 2 molecules of NADH. Everything that is circled in green is what you need to take away from step 1, glycolysis. So let's go back to our PowerPoint and watch a bit of a video. Now, I don't think it plays the sound, but this is included in your lecture. This is just going to show what happens during glycolysis. And again, remember they use these little black circles to represent carbon. We used those C's, same thing. All right, so there's our kind of animation of glycolysis. Next, some sources combine the next 2 steps into step 2. I'm going to break them in half and call it step 2A and step 2B. So let's go ahead and look at what we will call step 2A, the transition phase, or sometimes you will see it called the preparatory phase. And again, let's go back to our whiteboard. So step 2A is going to be the preparatory phase or the transition phase. And we're going to be looking at that pyruvate that we just produced. And that pyruvate that we just produced is going to go through twice. Or I guess a better way to say that is each of those pyruvates is going to go through the steps that we're going to draw out here. So everything that we draw right now on this phase is happening twice. Once to the pyruvate that we're looking at, and once to the other pyruvate that we also produced during glycolysis. So the pyruvate is now going to go through both of the membranes of the mitochondria. That outer smooth membrane is going to cross the intermembrane space, is going to cross the inner folded membrane and arrive at the matrix. And the moment that it arrives at the matrix, it's going to go through a very quick set of reactions to prepare it for the next phase. So this pyruvate, there's going to be a couple of things that happen, and I'm going to draw them one at a time, but in reality, all 3 of these things happen at once. So first, we're going to lose a molecule of CO2. So that carbon dioxide that we breathe out, this is where some of it comes from. One of these carbons leaves, and 2 oxygens are attached to it and also leave. So remember we started out with C6H12O6. we've lost some H's already to the NAD plus that came along. We're losing a C, and we're losing 2 oxygens. So we're losing quite a bit as we go through these steps. Well, that leaves us with a 2-carbon compound. And the 2-carbon compound is going to have some electrons stripped from it. By who? NAD+. Remember, NAD+ always does the same thing. He comes in, he takes some electrons, some protons come along for the ride, and he leaves as NADH. And that bit of step there produces something that's kind of unstable. This 2-carbon compound is a little bit unstable. We want to make sure that he stays together before the next step happens. So we need a chaperone. Who did we say in our previous lecture was a good chaperone? Coenzyme A. Coenzyme A. He's going to come along and attach to this 2-carbon compound. And he's just going to hold that compound together until the next step. This 2-carbon compound with the coenzyme A attached to it is called acetyl-CoA. Acetyl-CoA. And that's the end of the preparatory phase, or the transition phase. So what do we need to know here? We need to know the name and where it occurs. It's the transition phase, or the preparatory phase. It happens on arrival at the matrix. So before we really fully get into the matrix, it's just as soon as we arrive. We start with a molecule of pyruvate times 2, because remember, the same thing is happening to that other pyruvate that we produced. We end with an acetyl-CoA times 2. We produce a waste carbon dioxide times 2 and an NADH times 2. So everything that we see here in black that I've circled in green happens twice. Once to the pyruvate that we just followed, and once to the other pyruvate that we produced in glycolysis. So this acetyl-CoA is going to enter into the next major set of reactions. But since we have 2 of them, well, everything that we draw on the next whiteboard is going to happen twice also for each glucose that we started with. So here is our acetyl-CoA that we just produced. We're going to follow what we call the Krebs cycle, or the citric acid cycle. This is step 2B. And now we are fully in the matrix of the mitochondria. So this set of reactions occurs in the matrix. Now, as we enter into the matrix fully, the acetyl-CoA is bringing in his 2-carbon compound. There is a 4-carbon compound called oxaloacetic acid, or oxaloacetate, kind of waiting at the door. And like a good chaperone, this coenzyme A is going to pass this 2-carbon compound off to oxaloacetate, and then the coenzyme A will leave to go do the exact same thing again. What happens to that 2-carbon compound? It is attached to the oxaloacetic acid to produce a 6-carbon compound. This 6-carbon compound is not glucose. It just happens to have 6 carbons. Remember, we have lost some hydrogens. We have lost some oxygens. This 6-carbon compound is called citric acid. And that's why the other name for the Krebs cycle is the citric acid cycle, because the first thing that we produce is citric acid. Now, there's going to be a lot of stuff that happens on this whiteboard. Some of it is going to be important, some of it not so much, but I'm going to draw it all so that it helps make it make sense. Anything that is important, we are going to come back and point out with our green color. And anytime you see me list a name, such as oxaloacetic acid or citric acid, you do need to know that. The other compounds that I do not write a name for, they have names, you just don't need to know them. So don't worry about the ones that I don't give names to. So the citric acid is going to have some water removed from it. That's where some of our water comes from that we saw in our equation for cellular respiration. And then all of the carbons and oxygens and hydrogens are going to kind of rearrange, but it's still a 6-carbon compound. You don't need to know the name for that one. Don't worry about it. But once we're in that stage, NAD plus is going to come along, take some electrons and some protons, and leave as NADH. Once that happens, we're going to lose a carbon dioxide. And if we've lost a carbon dioxide, what do we have? We have a 5-carbon compound. This 5-carbon compound is going to have some electrons and protons taken by NAD+. And when that happens, we lose another carbon dioxide as waste, and we're left with a 4-carbon compound. This 4-carbon compound is going to have our chaperone come along, coenzyme A, because this next set of reactions is going to be a little bit tricky and it's going to make some things unstable. So our coenzyme A attaches. We're going to go through some reactions where you don't really need to know the details. They're a little bit tricky. But what I will tell you is we gain a molecule of ATP. So we produced an ATP. Don't worry about how. It is through substrate level phosphorylation. The steps of it are not so important. When we come out the other side, the coenzyme A will let go, and we're back to just a 4-carbon compound. Once we come out the other side and coenzyme A lets go, FAD is going to show up for the first time. And FAD behaves just like NAD plus does. He's going to come along, take some electrons and some protons to become FADH2. When he does that, we still have a 4-carbon compound. No carbon dioxide falls off right now. However, we do lose some water. When we get to this phase, NAD plus is going to take some more electrons and protons to become NADH. And finally, after NADH is produced, what we're left with is oxaloacetic acid. And oxaloacetic acid is now in place to meet the next acetyl-CoA that shows up. And we can see that we've got kind of a big circle here. We just went in a cycle. That's why this is the Krebs cycle, or the citric acid cycle. This just keeps going. but everything in this one circle happens to this one molecule of acetyl-CoA. But we go through this entire circle twice, because remember, we have two acetyl-CoAs that came from our original starting glucose. So let's see what the important takeaways are from the Krebs cycle, or the citric acid cycle. You need to know the name, that it is step 2b, and that it takes place in the matrix. You need to know that we started with an acetyl-CoA. We produced citric acid at the start. We have two waters. I'm going to go ahead and write that up here. Two waters as waste times 2. One for each, or 2 for each acetyl-CoA. We produced 1, 2, 3, let's undo that, 3 NADHs. But again, times 2. We produced 2 carbon dioxide waste products times 2. We produced 1 FADH2 times 2, and an ATP times 2. What we were left with was an oxaloacetic acid. And again, times 2. So I'm going to go ahead and write times 2 over here with our citric acid. Okay, so make sure to know everything that is in green here. I guess I'll go ahead and write times 2 next to our acetyl-CoA that we started with. Everything that you see on here happens twice for our original starting glucose. So let's go ahead and watch another video that will show both our transition phase and our citric acid cycle. So here I've shown you that it's happening twice. But let's watch our video here. Okay, so now we're going to look at something completely different as far as how we draw it out, we're going to look at something called the electron transport chain. So let's go ahead and go back to our whiteboard and look at the final step of cellular respiration. And rather than drawing chemical reactions, this is going to be more of a step-by-step through kind of a diagram. And let me explain before we get going exactly what I've drawn here. This blue line across the middle of the screen represents that inner mitochondrial membrane. This is the one that's folded up into those cristae. Below the blue line is the mitochondrial matrix where those last couple of reactions took place. These kind of pinkish purple boxes are various proteins, different types of transport proteins along this inner mitochondrial membrane. This weird looking thing down here at the end is a very specialized protein called ATP synthase. You don't need to know the name, but that's just an important one that we're going to be looking at. Now, down at the bottom in the matrix, we see all of that NADH that we've been producing and all of that FADH2 that we've been producing. Because even though it looked like we were following the carbons, they were not the most important players. The important players were those guys that were coming in and grabbing electrons, because the electrons are really what we were after through all of that mess that we just drew. So the NADH and the FADH2 are all gathering down here in the matrix. We also have some of those extra protons. Remember when we made NADH, there was also that extra H+ that was generated? Some of those are down here in the matrix also. Above this inner membrane, we see another blue line. This is that outer smooth membrane. And above that would be out in the cytoplasm. In between those two membranes is the intermembrane space. And in the intermembrane space, there are a lot of protons. The number that I've written here is not significant. I just did this to show that there are a lot of protons there. Also down here in the matrix, kind of gathered near this third box here, is something written as 1/2 O2. So I'm going to talk about why it's written that way. And I wrote it that way because that's how your book writes it. That's how most book writes it. But what is 1/2 O2? 1/2 O2 is O. But why did they write it this way then? Well, as we saw earlier, O2, molecular oxygen, that's what we breathe in. It is two oxygens joined together. By the time we get to this point, that molecular oxygen is split in half into an O oxygen and another O, another oxygen. But they show it as 1/2 O2 because they want you to realize, where did this oxygen come from? It is from the air that you breathe. And finally, this weird looking guy over here, gathered near the bottom of it, are a lot of ADP and a lot of inorganic phosphate. So let's look to see what happens at the end of cellular respiration. Up to this point, how much ATP have we made? We've made 2 net ATP in glycolysis. We made 4, but we spent 2, so we had a net gain of 2 ATP. In transition, we didn't make any ATP at all. In Krebs cycle, we had 2 ATP, 1 for each of those acetyl-CoAs that came in. So really, up to this point, we have made 4 ATP, and 4 ATP is not a lot. And all of the ATP that we've made so far was through substrate level phosphorylation. We're going to make significantly more ATP here using oxidative phosphorylation. Let's see how that happens. Well, that NADH and FADH2 that we've been following, that we've seen coming along and taking electrons, when they get close to this first weird-looking complex of proteins, something's going to happen. Remember, NADH and FADH2, they're electron carriers. They like electrons. But this weird complex of proteins likes electrons more. So when NADH gets near this first box here, any electrons that he's carrying, this protein is going to take them. If FADH2 gets nearby, then this weird little extension is going to take them and pass them to the larger protein there. So this first protein likes electrons more than NADH and FADH2. Once we have some electrons inside this first protein, what happens? This protein has a negative charge. And what's attracted to negative charges? Well, positive charges. So any protons that NADH or FADH2 are carrying, or any of these free protons that we have down here, the protons are going to be attracted to those electrons. All right. Now, let's see, this second protein here, he likes electrons even more than this first protein does. So these electrons that are in the first protein are going to be passed or taken by the second protein. And once the electrons leave the first protein, there's no need for this proton to be there anymore. But the way that this protein is shaped, protons can enter from the bottom, but they cannot go back out through the bottom. And it's shaped so that they're not able to follow the electron as it moves from the first to the second protein. The only way out is through the top, into the intermembrane space. And that's why there are so many protons up here. Now, once we have this electron in the second carrier here, any protons that did not follow the path into this first protein, well, they're going to be attracted to that electron in the second protein. Now, when there is an electron in this second protein, the third protein down here is going to notice it, and well, the third protein likes electrons even more than the second protein does. So he's going to take it. And the same thing happens here. Once the electron's no longer there, the proton is going to get kicked upward into the intermembrane space. Now we have this electron in the third and final carrier protein. But we need to get rid of that electron. If the electron stayed here, well, it would get full. And then nothing would be able to pull the electrons out of the second protein, and he would get full. So we need to get rid of this electron. How do we pull it out of this protein? We need something that likes electrons even more than this protein does. Well, that's why we breathe. And this is the only reason why we breathe oxygen. And as I alluded to way back in chapter 2, this is the only reaction in the body that we want oxygen for. Every other place in the body, free oxygen is deadly. But we need it for this one single reaction. Because what did we learn about oxygen way back in chapter 2? He's pretty far to the right. He's pretty high up on the periodic table. What does that mean? He's very electronegative. He likes electrons. We called him an electron hog. So he gathers near this third protein, and any electrons that are there are going to get pulled out and attach to that oxygen. That oxygen is gathering electrons. It's the job of the oxygen there, sometimes you'll hear him called the final electron acceptor. It's his job to pull the electrons out of the electron transport chain. So what we end up with is a lot of oxygens with negative charges. So we have a situation now. We have a lot of positive charges up here and not a lot of positive charges down here. So we have a couple of gradients. We have a concentration gradient just because there's more of them in one area than the other. They want to come through. But also we have a lot of positives up here and a lot of negatives down here. So we have an electrical gradient. When we have both a concentration and an electrical gradient in the same direction, we call that an electrochemical gradient. And that's what we have here. These protons really want to come down here and attach to that oxygen. But what did we just say? These are all one-way passages. They can't come through. And these membranes here, membranes don't like to allow positive charges to pass through them, or really anything with a charge to pass through them. That's why we need carrier molecules if something with a charge wants to pass. But these carrier molecules, they won't let these protons pass through in the other direction. That's where this last protein comes into play. This last protein, the ATP synthase, does let H plus pass through. And it passes through a lot. These protons are just flooding through. And when they come through, they're going to attach to these oxygens. And for every oxygen, we're going to have 2 H pluses attached to it. If we have 2 H's and an O, what do we end up with? H2O, water. And this is where more of that water comes from that we talked about earlier. Now, this protein is shaped pretty weird. One, it's a tube. It lets the H pluses pass through. But down here at the bottom, every time an H plus passes through, a couple of things happens. It spins, and one of these weird little kind of almost like doors that I've drawn here at the bottom, they kind of kick forward. And as it spins and kicks forward, it takes an ADP and an inorganic phosphate and slams them forcefully together. So the ADP and the inorganic phosphate, if they're slammed forcefully together, what do we end up with? We end up with ATP. Now, for every 1 molecule of glucose that we started the entire process with, this step right here, this oxidative phosphorylation to produce ATP, we get about 28 molecules of ATP. About 28. This is where all of our ATP comes from. We got 2 in glycolysis, we got 2 in the Krebs cycle, but we get 28 from the electron transport chain. So all total, we got about 32 from 1 molecule of glucose. So what are the important takeaways here? This one's going to be a little bit trickier to circle because of the way that it's all drawn out. But we got 28 molecules of ATP. It's step 3, the electron transport chain, and it happened along the inner mitochondrial membrane. Okay, now you do need to know the steps, so I'm not going to go through and circle every single thing on this one, but you do need to know what's happening in this picture. All right, now let's go back to our PowerPoint here. This is a variation of figure 26.8 in your textbook. This is from a different source, but it's still showing the same thing. Here are all of our NADHs and our FADH2s. Here are those proteins along this middle or this inner membrane. Down here is the matrix. Up here is the intermembrane space and the outer membrane. And right down here is that weird shaped protein, the ATP synthase. Here's our ADP and our inorganic phosphate being slammed together. Now let's watch a quick video, and again, this is attached to the PowerPoint, so just listen to it on your PowerPoint, but this is going to show everything that we just drew all the way from the beginning. So this is showing the entire cellular respiration process. So here kind of sums up everything that we just talked about. Up at the top, glycolysis, we netted 2 ATPs, we produced 2 NADHs and 2 pyruvates. Each of those pyruvates goes on to the transition phase or the preparatory phase. We got some CO2 out of it, we got some more NADH plus or NADH. We entered into the citric acid cycle. From the citric acid cycle, we got more NADH, we got more ATP, some FADH2s. And then from the electron transport chain, all of those NADHs and FADH2s entered. We got a lot of ATP, some from the NADHs, some from the FADH2s. Don't worry about how much ATP from the FADH2 or how much from the NADH. Don't worry about that. That's not important. And then at the end, we got water and a lot of ATP. So we've all heard someone say, feel the burn when they're working out, or if you've ever, ran or did any sort of physical activity that you had to exert yourself, if it lasted long enough, you started feeling kind of a burning sensation. And that feel the burn is kind of a common phrase that we're all used to hearing. So what is that burn? Well, when you are using oxygen to produce ATP at a faster rate than you are replacing the oxygen, that's going to tie into something that I said just a little bit ago. Let's go back to our picture here. If you are using oxygen faster than you are replacing it, then we do not have enough oxygen kind of down here. And if we don't have enough oxygen, then we don't have anything to pull the electrons out of this third protein. And eventually this third protein will get full of electrons. And if he is full of electrons, then he's unable to pull electrons from this second protein. And if he can't pull electrons from the second protein, then this second protein gets full and can't pull electrons from the first protein. And if this first protein gets full of electrons, then he can't pull electrons from the NADH and the FADH2. So NADH and FADH2 become occupied, they become full. So if they're full, then basically the electron transport chain stops. And if the electron transport chain stops, then we have no way of kind of proceeding with the citric acid cycle. All of the NADH and FADH2s are occupied, so we can't keep this going. And if the citric acid cycle stops because the NADH and the FADH are full, then the preparatory phase also stops. The only thing that can still occur is glycolysis. Glycolysis can still occur in the absence of oxygen. But the prep phase, the Krebs cycle, and the electron transport chain all stop. And that's because NADH and FADH2 become full. Now, one thing that's different in glycolysis is if we have all of those other phases stopping, we actually take this pyruvate, and instead of passing it on to the preparatory phase, it gets converted into something called lactic acid or lactic acetate. I'm sorry, lactate or lactic acid. And lactic acid is starting to build up in your cells, especially in muscle cells. And like any acid, it's going to lower your pH. Lowering your pH is going to result in a burning sensation. And that's where that feel the burn comes from, because of a buildup of lactic acid. And the reason that the lactic acid is building up is because of inadequate oxygen. Now, before we talk about disruption of ATP production. I want to go back and look at the equation for cellular respiration. We've now talked about cellular respiration all the way through. So here is our equation. Here's our C6H12O6. Where did that come from? From the food that we ate. That's what we just followed. That's what we started with in glycolysis. There's our oxygen we breathe. That's the only reason that we breathe oxygen, and that's the only thing that our oxygen that we breathe is good for. Here's our arrow that represents everything that we just drew. And along the way, what did we do? We produced water as a waste product. We produced carbon dioxide as a waste product. We got a lot of ATP out of it. We got about 32 ATP. And heat, because conversion is inefficient. So hopefully that kind of big, scary looking equation makes a little bit more sense at this point. So there are ways to disrupt ATP production, and that's not something that we want to happen, but it's something that is possible to happen, and various metabolic poisons can do that. And there's a lot of different types, but let's look at two of the more common. Cyanide. Well, cyanide is what we find in apple seeds. If you've ever been told apple seeds contain poison and they'll kill you if you eat them, well, there's a little bit of truth to that. You would have to eat pretty close to your weight in apple seeds before they do you any real harm, but there is cyanide in apple seeds. But cyanide is actually also what's used in the gas chamber. It's potassium cyanide, but it's still based on cyanide. So how does cyanide work as a poison? It actually interferes with one of those protons in the electron, or those proteins in the electron transport chain. And if you interfere with those proteins in the electron transport chain, then what you do is you block the movement of electrons through the electron transport chain. And if those electrons aren't moving, then we're not getting that big payoff of ATP at the end. Another class of these poisons are called uncouplers. And uncouplers poke holes in the inner mitochondrial membrane. Well, why is that important? Let's go back to our drawing here. If we had holes poked in the electron transport chain anywhere through here, well, the protons now have no reason to go through that ATP synthase. Instead, they could just go through any of those holes. And if you're not going through the ATP synthase, then we're not generating ATP. And finally, kind of outside of cellular respiration, let's talk about how our body handles glucose, how we store it, how we handle when we have too much of it. So when we have glucose demand, that glucose is going to move into cellular respiration. But if we have glucose and we're not needing it at the moment, if we have excess glucose, we do not lose that glucose in the feces, and typically we don't lose it in the urine, although if we have way too much, sometimes it shows up in the urine. So what do? Instead, what we do, instead of getting rid of it, we're going to store it as something called glycogen. And what we can see is we have a molecule of glucose, another molecule of glucose, another molecule of glucose, and so on and so on. And we just attach one to the next, to the next, to the next. And we end up with this long branching chain, a lot of glucose stuck together, and that's called glycogen. We mentioned that back in chapter 2. Glycogen is stored a few places in our body. It's stored in the liver. It's stored in some of our muscles, it's stored in the heart. And when our body doesn't have adequate glucose, then what we can do is actually take this glycogen and break these bonds. And when we break the bonds, individual glucose molecules break off, enter our bloodstream, and our body can use that for cellular respiration. So if we have glycogen, where does that glycogen come from? It's taking one glucose and another glucose to stick together, and another glucose sticking it together, and another glucose sticking it together, making glycogen. Well, that process is called glycogenesis. Genesis means formation of. Formation of what? Formation of glycogen. So glycogenesis is forming glycogen from glucose. And what about this second process where we take the glycogen and we break it apart to free up some glucose? That's called glycogenolysis. Remember, lysis means to split. Splitting what? Splitting glycogen. So glycogenolysis is making glucose from glycogen. Well, we can get glucose some other places. Sometimes we can break down fat to produce something like glucose, some form of carbohydrate. Or sometimes we could break down proteins to produce some form of carbohydrate. So what do we call it when we're producing glucose or a carbohydrate from something other than a carbohydrate? That's called gluconeogenesis. Gluco for glucose or carbohydrates. Neo, new. Genesis, again, formation of. So formation of new sugars. Gluconeogenesis is the formation of carbohydrates from non-carbohydrate sources, such as fats and proteins. Formation of carbohydrates from non-carbohydrate sources, such as fats and proteins. Now, this can be a good thing. Say you're working out and you're losing weight, you're breaking down excess body fat. But this can also be a bad thing if, say, you're starving to death, or if you have certain types of diabetes, your body might start consuming itself because you don't have adequate fuel. So we have different ways of handling glucose, storing it or accessing it based on the body's needs. All right, I know that was one of our rougher lectures so far. Students either really like that lecture or they really hate that lecture. So if you fall into that latter category, if you did not like that, if you didn't feel like you understood it, watch the lecture a few times, ask questions. I'm happy to help you. Our tutors are happy to help you. Just let me know. Don't put it off if you don't understand it. Ask early. All right, take care, and I will talk to you next time.